Yes. Yeah. Hi, everybody. Thank you so much for being here, a part of this webinar. I'm excited to be with you all and to discuss this topic of selecting the right antibody. Now, I must say, the way my screens are set up, I cannot see if people have questions. So I think maybe just like our host said, if we just want to address everything at the end, that would probably be best. Well, just a quick introduction on my part. So I have been with Santa Cruz Biotechnology since 2018. I was brought on to be a part of the tech support team, which is a team that troubleshoots our antibodies and products with researchers if they have questions. Um, handling our antibodies if they are doing an experiment, say Western blot or immunoprecipitation or one of the various assays that you can use antibodies with, then they'll call our department and we basically try our best to help them through their experiment to get to the best possible result. And so I was a part of that team for about 4 years, and then I got moved over to the chemical side of things, where I'm now a supervisor for our chemicals and licensing department. But during the course of the 4 years that I was a part of the tech team, I had a ton of phone conversations and video conversations with customers and researchers about antibodies, about selecting the right antibody for their research and just helping customers get the best possible results. So yeah, it's been a pleasure talking about antibodies. It's definitely something that I have really enjoyed over the past few years. So I'm excited again to be talking about antibodies with you all today. I think we can start first by just talking about kind of an overall objective. So overall objectives here, to learn how to effectively select the correct and best antibody for the experiment, right? Before we begin, we have to ask a simple question, okay? And that question is, what am I trying to do with my experiment? Or what is the researcher trying to do with their experiment? What is the end goal and what's the hypothesis, right? Like what is the main thing that they are trying to do with their research? Okay, because that's going to play a big part in how we eventually select our antibody. Okay, so I just kind of wanted to put that up just to highlight the main objective here. Sure, it's to select an antibody, but we can't even do that unless we know what the overall objective is of the research. So let's dive into part one, which is selecting the protein of interest and the sample. This is going to lead us in to selecting the antibody and some of the upcoming parts. So selecting the protein of interest, this is also known as the POI. Okay, so we must talk about this, okay, because it's extremely important. Our protein of interest, this is essentially the target that we are wanting to detect in our experiment. Now, the type of antibody that we select, it heavily depends on the protein of interest. Okay, so we can't even select an antibody unless we know what protein it is that the researcher is interested in. Okay, so suppose you are trying to detect a protein that has some sort of interaction with various cancer proteins or carcinogens. That could be the goal, right? Maybe you could say, I want to know if this particular protein, say protein X, has some sort of interaction with other carcinogenic proteins. Okay, and it could be anything. I'm just saying X just to highlight. It could be anything. I mean, there's tens of thousands, if not hundreds of thousands of different proteins, right? So, I mean, the options are almost endless if you think about it. This is going to be something that you need to get from either the researcher or whoever is doing the experiment. They need to tell you what their protein of interest is, right? So say again, you want to know if there are any levels of carcinogenic cells or proteins in your sample that you're working with. You'd think to yourself, well, if I measured the amount of tumor suppressing proteins in my sample, then this can indirectly tell us the presence of carcinogenic material or tumors in our sample. Okay, so this is just kind of like a thought, right, that a researcher might have. And I'm being broad on purpose because, again, it's hard to be specific in these areas since we could, research is so broad. But you could ask this question, right? You could say, all right, well, what is a highly researched tumor suppressing protein? Well, a really, really highly searched tumor suppressing protein is P53. It's also actually called a tumor suppressor protein P53 or TP53. Okay, so we could say, all right, well, here's what I'm wanting to do with my research. All right, so now I have basically a protein of interest. My protein of interest is this P53, this tumor suppressor protein. So we have our protein of interest based on what the overall goal is, and that is, again, to just study interactions between carcinogenic material. But we also have to think about what species that we want to test in, or what the researcher wants to test in. Oftentimes in research, it is mouse, rat, or human. And there are various other species that researchers could test with, but these are the big, main three. So let's just say, for sake of simplicity, suppose you pick human. Okay, so far we have our protein of interest, which is P53, and we have the species that we want to test in. It doesn't quite end there, though. Okay, because we also have to decide on what cell line that we want to test in. And again, this is just going to be something that you have to talk to the researcher about and ask them. Oftentimes, that's what I always do, is just ask them, okay, what cell line are you wanting to work with, or what cell line are you working with? And this is extremely important because for the application that the researcher is eventually going to do, they're not going to get very far in their results. They're not going to have really any good results unless they are using a cell line that endogenously or natively expresses your protein of interest. Right? So a tool that we use over here in the States is a tool called the Human Protein Atlas. Now, I'm not sure exactly which tools that you might have, but I'm sure that there are some sort of tools that you could use, maybe NCBI or some other ones, research tools, where you could actually search cell lines, and it could be human cell line, mouse cell line, right? All of these different things. Typically what we do is we just get on the Human Protein Atlas and we type in right here in the search engine P53. In fact, I know this is being recorded, but I think it's okay if I just click on this here. Let me bring this over. Here we go. So this is just kind of an example, right? So you can type in P53. Again, this is our protein of interest. Okay, so here's this first one. It's the first one that comes up, P53 or TP53. And you can go to cell line, right? And then what's really cool is it kind of shows us what types of cell lines, what types of cancers that this endogenously is expressed in. So this is just kind of a really good tool that we use over here. And I'll just take kind of one as an example, like HepG2. This is a cell line that is very easy to acquire. And this right here, it's showing that there's a kind of a medium amount, a medium to moderate amount of this protein of interest, P53, that this is expressed in. And again, since it's so easily obtainable, HepG2 would probably be a pretty good selection here. Now, some of these others, it's more expressed, but HA7 might be harder to actually obtain. Same thing with SNU423. But HepG2 is a really safe option. So we're just going to use that one for the sake of simplicity here. And I'll bring this back over. So what does this mean? Okay, and I know I'm kind of going by fast. I just want to get a foundation before we actually talk about selecting the antibody. So at this point, what do we have? We have the protein of interest, we have the species, and we have the cell line. So now it's time to actually select the application that we'd like to use, or the researcher would like to use to detect this presence of the protein of interest. Okay, so again, if you are the researcher, this means that you have to make this decision based on what you are wanting to do. Are you just trying to detect the protein of interest, or are you wanting to detect and understand where it's located in the cell? Because depending on what you are wanting to do kind of depends on the application that you're going to select. Right? But if you were just talking to a researcher, this is going to be something that you need to ask them. Okay, are you wanting to do Western blot? Or are you wanting to do immunofluorescence? One of these applications. So here's just kind of some examples, some of the examples that we often see with using antibodies. Western blot, IF, IHC, ELISA, and immunoprecipitation. Right, so here's kind of some pictures here. This is Western blot. This is immunohistochemistry. So these are like the big ones, right, that people use in order to use these antibodies. And again, it's just something that you're going to have to ask them before you can really start helping them decide which antibody to select. So again, this is just going to be the researcher's preference. I do want to highlight that without getting this last piece of information, it is hard to actually provide any guidance for selecting the right antibody. Now, I want to keep this example moving forward, just keeping on the same example that we've been talking about with the P53. So let's just say that the researcher wants to perform a Western blot. So here's what we know at this point. We know that our protein of interest is the P53. We know that the species that the researcher is wanting to work with is human. We know that the cell line the researcher is working with is HepG2. And that's, again, we're just kind of picking these for the example. And then the application is Western blot. Now, with all of this information, these are kind of the four main pieces of the puzzle that we need in order to actually select the antibody. So we can finally go through that selection process that's best suited for the researcher's needs. All right, part 2, selecting the antibody. So just, I have some kind of, just like an overview of antibodies very quickly here. So there are 5 major immunoglobulins. Immunoglobulin, of course, is just a fancy term, a scientific term that means antibody, right? So there are 5 major antibodies that have specific structures and functions. We're really only going to focus on one of them today, which is IgG. I will put all of them up here just for the sake of having more information. But IgG is definitely the most common type of antibody. It is the most common antibody that you will see in research by far. You'll see IgAs and IgMs somewhat moderately. Now, in my experience, I haven't really seen too much of IgE or IgDs. Most of the time it's going to be IgG, right? So this is found in the blood and in other body fluids, and its sole purpose is to protect against bacterial and viral infections. Okay, and then let me just move forward here. Okay, antibody structure. So antibody structure, IgA, IgM, and IgG. I wanted to talk about these very briefly, simply to point out the differences in their structure. So IgA and IgG and IgM, these are the big three, right? What's interesting is IgG and IgM can actually kind of work together as far as like primary antibodies and secondary antibodies. IgA is a little bit more difficult to work with. And there's something to do with this part here. I know this isn't a very scientific or super accurate drawing here, but I think it does a good kind of simplified representation of what they look like. There is something here that actually causes a type of hindrance almost that prevents IgGs and IgAs to work together. But usually even still, if you have a primary and secondary antibody, you want to typically keep them IgG. But just for the sake of, again, providing more information, IgGs and IgMs can work together. So anyways, I just wanted to throw this in here. I think it's kind of a good overview, just something quickly to talk about. Typically, you're going to see this as a Y, right? So kind of this structure here. When you see an antibody, they typically take this IgG form. Instead of like one of these crazy IgM pentamer structures or the IgA, okay? So when you see a structure like on Google or I don't know, just if you're looking at animations, they're always going to use this kind of structure, which is the IgG. Now, Santa Cruz, just kind of a little bit about our product line. We have several different product lines, from gene modification products to chemicals, right? But antibodies are easily the biggest seller, and it drives the most sales for the company for sure. Now, we only sell monoclonal antibodies. 99% of them are produced in mouse, and there's a very small select few that are produced in rat. And for those of you who have kind of been aware of Santa Cruz, we used to sell polyclonals many years ago. In 2016, we actually switched over to mouse monoclonals just for like greater specificity. And we offer several different versions of antibodies. So we call these conjugated forms. And we'll kind of talk about these in the next slide, but I just wanted to put them up here. So these are the various conjugations that we have to offer. And I'll also talk briefly about, I'll kind of show you a product page momentarily, that just because we have a conjugation available doesn't necessarily mean we have it available, you know, for all of our antibodies. For instance, this TRIT-C, this is a type of fluorophore. We might have it for some of our more popular antibodies, but we won't have this available for all of the antibodies possible. So it's just kind of one of those things you have to check the website for. And then I wanted to show you this. In fact, I think I might already have it pulled up on this other screen. Here we go. So P53, right? This is our, the P53 is the protein of interest. So here's the antibody that we are working with. Of course, this is just kind of one of the various antibodies that we have, and we'll get into that here in a second. But I wanted to show you the different conjugations, right? Kind of where to see them. And again, it might look different on y'all's websites, but just kind of showing you what exactly we have here to offer. So yeah, just wanted to show that very quickly. And to go over these one by one, I'll do it quickly, but I think it's important to explain what these conjugated forms are for. Because if you understand what the conjugates are for, it's going to help you make better recommendations for customers. It's going to help, if you're a researcher, it's going to help you understand kind of which ones that you might need for your research and so on and so forth, right? So we have our X form. This is a form of antibody that's basically 10 times more concentrated than the normal version. It's also gelatin-free, which is pretty nice for a multitude of reasons. We have our agarose conjugate. This agarose is typically used for immunoprecipitation. So you see that a lot for IP. Then we have our HRP. This is horseradish peroxidase, used for Western blot, ELISA, and immunohistochemistry. Then we have FITC, which is fluorescein isothiocyanate. This is also used for IF and flow cytometry. We have PE, it's phycoerythrin. It's used for these here. Now, these next few ones, I'm just going to click through. They're basically fluorophores, and these signify the wavelength. Okay, so wavelength 488, wavelength 546, so on and so forth. So yeah, you'll see these a lot for RGB Western blot. This is the type of Western blot that you'll see that has the different colors. You'll also see these used a lot for immunofluorescence and flow. Then we have biotin for immunohistochemistry, and then we have TRITC. This is also a type of fluorophore. And then we have Alexifor 405, PerCP, and then PCP C5. So I know it's a lot. I know it's a lot of different kind of options to try and remember. I think the best way to remember this is kind of just highlighting the big ones. HRP is going to be one of the biggest out of all of these, and it's because it's one of the main enzymes used for Western blot, but it's also used for ELISA a lot and immunohistochemistry. So this one is very, very common. So this is a big one. FITC is pretty big, and actually from FITC all the way down through these Alexaflors, all of these are pretty popular. We don't really see the PCP or PerCP, like we don't see these that often, but these are the ones that really shine for sure. Okay, let's continue on with our example. Really quickly, I wanted to show you these Cruz floors. This is just kind of like something that Santa Cruz offers. It's like an in-house brand, same type of stuff. They're just fluorophores that we develop in-house. I just wanted to throw this up here very quickly to let everybody know that we do have them and offer them. But the main reason why we're here, selecting our antibody, right? So as we discussed at the beginning, the first thing we need to know is the protein of interest. Well, we've already figured that out. That's P53, right? So let's go find a P53 antibody. Now, I understand, everyone might not be using our website, but I'm just gonna kind of show you the best way to do it on our website, since that's what I'm most familiar with. So what you can do is just come up here and type P53. P53 antibodies. Now, we offer tons of different antibodies, right? All of these are P53. So it's like, okay, just because we have our protein of interest, it doesn't mean that we immediately know which antibody to go with, right? That's why sometimes it's difficult for researchers to know which one is the best antibody, especially when you have so many different options, right? So the best way to think about this, and I'll go back to the slides. I'll kind of be jumping back and forth, if that's okay. So as you saw, we have so many different options, but remember, we are looking for human species, and we're looking for Western blot application. So the recommended assays are listed under applications. Right there, right? And then species as well. So going back to this here, typically it's going to be mentioned, but if it's not in here, say if you're struggling to find it, you can always go to the data sheet as well, and the data sheet should always have this information. Right, it's always going to have it here. It even says, TP53 human. So and over here as well as like applications and what you can use it for. So just more information, more detailed information is always going to be in the data sheet. So I always recommend to look at that. But it's not always necessary, right? So we can look at this again. We can say, okay, well, we know that we want to work with human. And we know that we are working with Western blot. Okay, so for instance, if we're going down the list here, human, 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 this one right here, we would not want to select because we only have it listed for mouse and rat and not for human. Okay, same thing with this one here. We don't want to use that one because it's not listed for human. And then same thing with this one here. Okay, so that kind of narrows it down a little bit, but obviously there's a lot of these where it's mouse, rat, and human. Human is included, but then Western blot is also included. So it's like, okay, well, I still have a lot of different options here. What's the best one to go with? So when you have multiple antibodies that have the desired specifications, such as species reactivity, application, all of these things, it's best to go with the antibody that has the highest amount of citations. And the reason why is because, think about it. I mean, this is going to be the antibody that has been proven basically to work in hundreds, and actually in this case, thousands of citations. So a lot of people have cited it. A lot of people have said it's a great antibody. So it's kind of like you want to go with the safest option. And the reason why we say this is because we understand that scientific research and these experiments that the researchers are performing can be very time consuming, and they can be expensive as well. So we don't want to waste anybody's time. We don't want to ever recommend an antibody that maybe doesn't have that many citations or has a poor rating or something like that. Now, if the researcher wants to make that decision and buy one of those lower cited antibodies, that's totally fine. They have the freedom to do that. But we always want to be more safe than sorry because we want to save the customer, the researcher. We want to save time for them and we want to save them money. And we also want to build a good relationship. Right? So it's like we have to pick the thing that matches the application and the species, and you have to make sure that you're selecting the antibody that detects the protein of interest, and you always want to pick the one that has the highest citations. Okay, so that's kind of like the main method that we would use in selecting the primary antibody. And notice I said primary antibody. We'll talk about secondary antibodies very soon. One last thing about citations, just wanted to put this up here. So we do have a section about citations, right? So sometimes researchers will actually, they'll go through these and they'll want to read the titles, maybe actually click on some of these. And these are all links, right? So it leads to NIH, PubMed, and you can read the abstract, and the researchers can do their due diligence in making sure that this is the best antibody for them. So that's one thing that they can do. It's just, again, one more thing I wanted to highlight. Now, occasionally, researchers will want to do research on phospho-specific antibodies. So these are essentially, proteins that have been highly phosphorylated. So they want to use antibodies that can still detect their protein of interest even after it's been phosphorylated. So let's take a look at P53 again. So if you, if we go back to the P53 page, if you keep just scrolling down, you'll see phospho-specific P53 antibodies. So you'll see all of these. These are the different phospho-specific. Anytime you see a P, dash, and then, followed by alphanumeric number, basically. So the P will always stand for phospho, right? And we would do this the same way. We would say, okay, well, the researcher is wanting human, and the researcher is also wanting Western blot. Let's go with the ones that matches those specifications, but also have the highest citations. And here what we get is this D9, and then we also get this FP3.2. Now, what's interesting, I like this example a lot because there's one more thing in this case that we have to look at, which is isotype. Okay, so this is a IgM, This is a IgG1. The reason why that's important is because depending on what your isotype is for your primary, this is going to kind of change how you're going to select for your secondary antibody, which again, we'll get into very soon. In fact, I think the very next slide we'll talk about that. But in this case, I would actually go with the IgG, even though it has a lower amount of citations, only because IgGs are so common and have so much research behind them. I typically like to go with IgGs just because it's it's well researched, it's the safer option, and I think it's probably just going to be more beneficial for the researcher. Typically the researchers will have IgG secondaries laying around and not IgM secondaries. So that's another reason why, if they have all the equipment like the secondary to do an IgM, that's totally fine. They can go that route, but they just have to make sure that they do have the right isotypes for the secondaries. So that's just something to kind of keep in mind. Now let's talk about that a little bit. Let's talk about choosing our secondary antibody. So just kind of a recap of how antibodies work. I'm sure everybody is at least somewhat familiar, but I just wanted to do this very quickly here. So we have our antigen, right? This is like our protein of interest. Then we have our primary antibody. That's what this yellow antibody is. Now we need a secondary antibody. That's what this green like upside down Y is. Now the secondary antibodies, they will typically have some sort of conjugate on it, right? That's what we've been talking about earlier when I said the alexiflors and the HRPs and all of those things. So that is the enzyme. Then you can come along, you can add your substrate and it gets your product, right? And that's going to be what your signal is. I just wanted to kind of bring this up because I think this helps visualize why we need a secondary antibody in the first place. So with that, now that we have this visual, let's talk about the compatibility between the primary and the secondary antibodies. So not all primary and secondary antibodies are compatible with each other. So how do you know which antibodies will work together? And more importantly, how do you select the correct secondary for your research? There's three main points here. There's three main ways that we can do this. So number one, matching the host species of the primary antibody. I'll talk about that in detail in just a moment. Number two, selecting the correct conjugate based on the intended application use. And then number three, defining the isotype and the subclass of the primary antibody. So let's break these down a little bit. Let's actually flesh these out. I think it's going to be beneficial to do so. So number one, matching the host species of the primary antibody. So our primary is a mouse monoclonal, so our secondary needs to be anti-mouse. Well, how do I know this? How do I know that our primary is a mouse monoclonal? Let's go back to this example of P53, right? Remember, we selected this one because it has the most citations, all of these things, right? So this one would probably be the best one. If you see right here, it says mouse monoclonal, and then it says IgG2A. And something else just to kind of remember, if you are getting an antibody through Santa Cruz, almost all of them are going to be mouse monoclonal, so it's going to be kind of easy to think of that anyways. But if you're not sure, you can always look right here in the description. Again, you can go to the data sheet here, and you can always find that information as well. So our primary is mouse monoclonal. So our secondary needs to be anti-mouse. So let's go look at our secondary antibodies. So as you can see, we have anti-rabbit. We have anti-goat, right? Anti-hamster, all of these things. Now, eventually we'll get to this point where for our secondaries for the mouse, we actually label them slightly different. We don't call them like, anti-mouse in the actual description here, but they are. You'll see right here, it says anti-mouse binding proteins. So a binding protein is it's somewhat different actually than a traditional secondary antibody. They're more of like recombinant secondary antibodies. They have the same exact function, to be honest with you. It's just since they're more like we've always engineered them as like a recombinant binding protein, but that's honestly not that important. I just wanted to highlight that anytime you see this BP, a binding protein is what that stands for. It's basically a secondary antibody. Okay, so you're going to see a lot of these binding protein, binding protein. These are all secondary antibodies that we offer. And then you can also see anti-mouse, 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 right? So let's go back to our slide. So we need an anti-mouse. So this means we cannot use any of these. We can't use the hamster. We can't use the anti-goat or the anti-rabbit. So all of those are off the table. So we can basically start with these. Now, what's our second thing that we have to look at? Selecting the correct conjugate based on intended application use. So we're doing Western blot, remember, this is the application that we're doing in our example. So we're going to choose a HRP conjugate. Now for staining such as IF, you would want to use an Alexaflor, or for IHC or ELISA, you could also use HRP. Again, it really just depends on what the researcher is wanting to do. But let's go back to our list here. So we want a anti-mouse and we want an HRP. Okay, so this is possible. So far, at least, based on what we've talked about, this is a possibility. That's not a possibility. None of these are possibilities. Oh, there's one. Maybe that one. We can't use any of these. We can use that one. So I think you kind of see the pattern here. We're looking for anti-mouse and HRP for a secondary antibody. But as you can see, we have options. We have 1, 2, 3, 4, 5, and 6 different options. Okay, out of those 6, which one do we select? And that's where we get into this final thing that we have to talk about, which is defining the isotype and subclass of the primary. So our primary antibody, let's go back. P53 is the one that we selected. So you'll see it right here, IgG2A kappa. IgG2A kappa. If you look at the list, you can also see it here, isotype IgG2A kappa. You can see the different isotypes here. The reason why this is important is because we typically want to match the isotypes. Okay, so we want to do this for the sake of being as safe as possible. Okay, they must be the same isotype in order for them to always be compatible. Now earlier you heard me say IgGs and IgMs sometimes can be compatible with each other. That is true. However, it's best to not take a risk, which is why we always say just try to pick the same exact thing. So if our primary antibody is an IgG2A kappa, then our secondary antibody needs to be the same. Okay, so we have an IgG. Now it's not 2A, but it is an IgG. We'll actually talk about this FC binding protein, but I'm going to come back to it. So we have this IgG1, so that's not quite the same. We have this IgG2A. Okay, that's a good sign. We have this IgG2B, probably not. We have the IgG3, probably not. IgG kappa. So that's a maybe. It doesn't have the 2A, but it does have the kappa, so that's a possibility. And then we have an IgG lambda. So this kind of helped us narrow it down between 3. We have this one, that's a possibility. We have this one, the IgG2A, that's a possibility. And then we have this IgG-FC. Okay, IgG2A. Now, based on what we're looking for the proper secondary, it's either going to be the FC binding protein or it's going to be the IgG2A. Here's the reason why. This last one here, this kappa, as you can see, look how many citations it has. It has a ton of citations, but through our experience, this one could absolutely work. Don't get me wrong, it could work, but this is actually a slightly better match because it's IgG2A. So this is the exact isotype, and I only know this basically from working with Santa Cruz. All of our generic binding proteins are already going to be kappa, which means this is a kappa. So this is an exact match. So this is probably going to be our best one. But I wanted to talk about this a little bit too. And I wanted to talk about it, antibody anatomy. Because I wanted to show you this. So you have a FAB and a FC region. This is also known as the heavy chain region. So whenever you're looking at these, this FC, it means that this is going to bind to that FC region. It's going to bind right here. Most secondary antibodies actually bind up here. The reason why this is important is because since this binds to the heavy chain, you actually don't need the subclass to be the exact same. It just needs to be an IgG. It doesn't have to be 2A or 2B or, whatever your primary antibody is, because it's not binding to the same spot where all of those others are binding to. Okay, so this is called a FC binding protein. It's a newer product, but we are starting to get more citations on it, and it has a really high rating. So I actually really like this. I'm a big fan of the FC binding protein. So anyways, you could use either of these, the FC binding protein or the IgG2A. Okay, so we have our primary antibody and we have our secondary antibody. The primary is going to be that P53 antibody, which is that SC126. That was the DO1 that we talked about. And then the secondary antibody is that IgG2A kappa binding protein. So now we are actually ready to perform our experiment, or now we are ready to help the researcher perform their experiment. Okay, so that's basically it. So I walked you through selecting or the proper antibodies, a primary antibody and secondary antibody. I have a very quick FAQ section that I'm going to go through, and then I can answer further questions after that. So I'm just going to kind of run through this here. Okay, so what if the customer wants to do a direct experiment? So the example that I showed you, we used a primary and a secondary antibody. That is known as an indirect experiment. Okay, a direct experiment means that you are you're just using a primary antibody. Okay, so if we go to our P53, all of these are directly conjugated. So this is a primary antibody, P53, and it already has the HRP conjugate, right? It already has this enzyme that's bound to the primary antibody. So you don't need a secondary antibody in that case. Okay, so you can, if the customer is wanting to do a direct experiment, you can just guide them to these directly conjugated antibodies. Now, what if the customer wants to perform ELISA instead of Western blot, right? Are there different antibodies that they must use? Not really. They would essentially use the, their primary antibody, and they can use, they could do this in a direct or indirect fashion. There is one thing that they need to use. It's called TMB. TMB is what it's called. We also have more information in our protocols. So you can always go to, ELISA, and it will say it in here. So we actually have alkaline phosphatase conjugated secondary listed here. We've discontinued these. So this needs to be updated. So I apologize about that. But this needs to be updated. So we would change this just to HRP. And then for the substrate, we would use that TMB instead of the PNPP. So this is a protocol that needs to be updated. But yeah, as far as antibodies go, though, no, they can use the same, they would just use HRP. Now, what if the customer wants a higher concentrated form of the antibody? Now, remember, we talked about this. So this is going to be the X form here. This is a 10 times concentrated form. So, this our generic form is 200 micrograms in 1 milliliter. This is 200 micrograms in 0.1 milliliter. And what's great about this is if the researcher wants to freeze the antibody, they can actually do that with this and only this form because it does not have gelatin. There's gelatin that comes with all of these other vials, and if they freeze one of those vials that have gelatin in it, it can actually damage and disrupt the antibody. But the X form does not have that gelatin in it, so if they do want to freeze it, they can use that form. I think that's actually the next one. Yeah, so they would use the X form here if they want to freeze it. Okay, and then this is the very last slide. What if the customer wants to do gene modification? Do they need antibodies for that? They don't necessarily need antibodies for the modification part. Here are all the gene modification products, right? The knockout plasmids, all of these, they're going to be gene modifiers. So they don't need a antibody for this part, but typically researchers will use antibodies for the following application. So in other words, they'll go ahead and they'll knock out their gene, and then after they perform the knockout, they'll typically go on to do like a Western blot, right? So if they're doing a Western blot after the fact, then yes, they would need to use a primary antibody, this 126 in this case, if they were knocking out the P53 gene, right? Okay, and then what if they're wanting to do capture or looking for capture antibodies for sandwich ELISA? So actually, believe it or not, we do not support sandwich ELISA. So if you ever have a customer that is wanting to get capture antibodies through SantaCruz Biotechnology, we don't have validated capture antibodies for sandwich ELISA. So just something I wanted to mention here. Sometimes we get that question. Now, what if the customer is wanting to use an antibody that reacts with a different species like zebrafish or sea urchin, canine, bovine, all of these, right? What do they do in those situations? Well, let me go back very quickly. Here we go. So the species are listed here, right? Mouse, rat, and human. However, there are way more species that are recommended like in these citations. Okay, and I think I actually remember seeing one in this A1 clone. Oh, it's not wanting to load. That's okay. I'll go to this one, see if this loads for me. Yeah, I don't know why it's not loading. Let me try one more time. I just wanted to show you this this citation. Oh, it's not loading. That's okay. But essentially, they can go through the citations. And a lot of times in the title of the citation, it will actually say what species that the journal was, published using. So in other words, it will say, P53 protein tested using zebrafish or using porcine or bovine. And it's just kind of a good source. So even if we haven't tested it ourselves, typically researchers will look through the primary literature to see if anybody else has used it with, zebrafish or sea urchin or one of these other things. And if they do find a publication where another researcher has used it for one of these other species, that can give them confidence on buying the antibody and actually using it for their purposes. And then what if the customer is wanting to do IP? Do they need any special antibodies? No, they don't need any special antibodies. However, they do need an agarose conjugate, right? We talked about this very briefly earlier. So they need an agarose conjugate if they're wanting to do direct If they don't want to do direct and they're looking more of like an indirect approach, then they can just get the unconjugated form, and then they can use one of our other products here, right here. These are basically agaroses, so they'll use the primary antibody with a agarose, and that's how they would perform their immunoprecipitation. So just kind of another option that they have. But that's basically it. Let me go ahead and back out of this, and then if there are any further questions here,